Okay. All right, everybody. We've got John Odenkirk here from the Virginia Department of Wildlife Resources. He works in the Northeastern District. Thanks for making it, John. We're going to turn it over to him. He's going to talk about brook trout and brown trout in the Appalachian mountain stream. All yours.
Cool. Thank you, Nat. Good morning, everybody. And that's the camera right there. So we're looking at the virtual people. Okay.
And do you have a clicker?
I have a clicker. Yes, I do. So yes, good morning, everybody. And thank you for having me here. I'm really flattered that somebody wanted to hear our talk. This work was presented at the SEFWA, the Southeastern Association of Fish and Wildlife Agencies. And it is always nice when somebody wants to hear what you found out about. And honestly, a little mea culpa here. This wasn't really a study. This work came out of a long-term monitoring program that we have on a couple of streams. There are sort of sentinel streams where we try to survey them annually to have long-term database on brook trout resources. And so this sort of fell in our laps, while we're monitoring decades-long data sets of these streams. One of the streams, brown trout, show up. It's kind of like having snakeheads fall in your lap, and then you just sort of have to, make hay out of it or make something out of it. So Mike Isel is my co-author on this, and once we watched this sort of thing occur over a number of years, we thought it was interesting. We thought it was exciting or different than some of the stuff that's in the literature, and so we wanted to disseminate it. We thought it was valuable information to put out there, and so we wrote up a nice pub and submitted it to the North American Journal, where it was promptly rejected. The problem was, in the editor's eyes, is we didn't have any replicants. We didn't envision this to be a ivory tower ironclad hypothesis testing experiment. This was sort of a case study, again, that fell in our laps, and we thought it was important enough to put out there. So after some changes, we resubmitted it to a different journal, Southeastern, which is now called Southeastern Association. No, it's the Southeastern Journal of Fish and Wildlife Agencies rather than the Proceedings. So some of you may have seen it or read it. Some of you may have seen the abstract. It's available as a PDF. If you'd like to see it and don't have it, I'll be happy to share it with you. So after all that, I will get on with this. Mike Isel and is my co-author on this. And we'll see if this works. It worked before. Went this way. How about if I just point it to you and you click it? The pointer still works on the wall, but it doesn't work on the screen. The clicker doesn't. Oh, did you do that or did I do that?
You did that.
Try again. So in Virginia, like many states on the eastern seaboard, our brook trout resources are in the mountainous areas in the western part of the state. I don't think that the Fairfax County distribution was accurate. We've had going back and forth with different people about that for years, but disregard that. Suffice it to say, though, that in the Rappahannock watershed, and this is an Eastern joint venture slide, thank you very much to the venture for that. The Rappahannock watershed, like many of these watersheds, historically had brook trout resources. The range is shrinking, obviously, and so this is not a good thing, and it's shrinking for a number of different reasons. And again, thank you to the Eastern Brook Trout Joint Venture for this information. And these are the threats, as you all are very acutely aware of, in order. And what was interesting to me when I first, was looking to see, okay, what are the greatest threats to brook trout? And so not only do we have non-native listed, and of course brown trout are non-native. So essentially, brown trout are fingered twice as the greatest evils standing in the way of brook trout conservation. Which I thought, okay, where's mackerel? Where's rainbow trout? What the hell? Well, they're non-native, so I guess they're covered there. But again, fingered twice. So obviously these things are really bad, right? And so in the Conway River, which is one of our sentinel streams, brown trout were discovered in 1987. They've been there before that, but we found out about them in 87. Stocked historically downstream and made their way up, and we're happy about it. Well, was this a problem? Initially, when I go trout fishing, I go to the Conway River because I enjoy catching brook trout and brown trout. Brown trout are typically larger. Anglers like that. So I'm not the only angler that liked it. Most of our anglers were very happy with this. Some members of TU were concerned that perhaps the brook trout would be taken on the chin. So we started doing a little historical literature research. What are the interactions, sympatric populations? And again, as many of you are very aware of, There's spawning interactions, potential reproduction interactions, feeding interactions, a lot of interactions that sometimes don't work out well for the brook trout. And I think this is, we're gonna come back to this a little bit in a minute. In some streams, the decline of brook trout in the presence of brown trout is when there's some additional factor, a stressor, whether it's a parasite, whether it's poor land use management, siltation, changing water temperatures, things like that. So you bring in something that maybe makes it a little harder for the brook trout to compete with a newcomer. So for the Conway River, this is the stream where the brown trout showed up. We have annual samples beginning in 20, and this was a subset that the editor wanted me to peel out, wanted us to peel out for the paper. So we could have gone back further in time, but we trying to make it more of a discrete data set. So 2011 through 2019 annual data sets, except 2018 was the wettest year in history, at least recorded history. Don't know about Noah's Ark, but prior to that, we had some sporadic samples. But we ended up with a data set about 15 years. Rapidan River, again, is a very similar river, adjacent watershed. The Conway River actually feeds the Rapidan below our sample sites. Very similar data set. But again, both of these are amazingly pristine forested watersheds. It's either Rapidan Wildlife Management Area, state-owned land, or national park, Shenandoah National Park. So our idea was to use the Rapidan next door as our reference stream to sort of make some plausible ideas about what might be happening. You're gonna have to, I'm just gonna have to point to you. I'm gonna set this down and point to you. Thank you. So in both rivers, we're doing our standard 3-run backpack depletion with 2 backpack units. We're reserving our fish after each run, and then thanks to Jack Van de Ventner, I hope he's doing well. We're using our beautiful little microfiche program to give us our biomass estimates and capture probabilities and cool things like that. And just again, watersheds are remarkably similar, haven't been disturbed in the better part of a century, except for a couple minor logging operations. Conway River is slightly lower elevation, slightly lower slope and gradient, but the next one. The drainage area, what you'll notice in some of the bar charts in terms of kilograms per hectare or number per hectare, the Rapidan is going to be a little higher. It's a bigger, a little bit more productive watershed, bigger stream, bigger width, etc. So that might explain a little bit of the difference, but other than that, very similar watersheds. Our typical gauntlet, almost all the fish are captured on run 1, very high capture probability. There's not many escapees. Some of you can recognize some faces there. Okay. So I'm going to throw a bunch of graphs at you. I've tried to differentiate for some highlight descriptors in the titles so we know what we're looking at. First, we're going to be looking at Conway. And most of this is going to be biomass. Biomass and numbers were very highly correlated. It seemed redundant to report both. But it's assuming that average size of adult brook trout didn't change in the Conway River with the brown trout water. So I've got a little bit more about that later. But so for now, we're going to kind of concentrate on biomass in terms of kilograms per hectare. And the y-axis, we got 60 kilograms per hectare as our maximum biomass. And what we see is the brook trout with the brown trout now have a record level in the most recent survey. And we're continuing the survey. We have data for 2020. It's not in here because obviously you have to cut it off at some point if you're going to try to publish it. But we have a non-significant increasing trend in brook trout biomass in the Conway River while brown trout colonized. Okay. We have a very significant increase in brown trout biomass in the Conway River over the same time period. It looked like they were kind of simmering for a while, and then conditions got right, and they got exploded, sort of like snake heads in the Rapidan. So what's interesting, I think, and the next slide shows it a little bit, is that this appears to be additive salmoniid biomass in these fairly infertile freestone headwater streams, which is, we didn't expect this. We thought it would probably be some level of compensatory trade-offs here, but for whatever reason, this biomass appears to be additive. So on the left side is total salmoniid biomass, and the right is percent of brown trout as a component of that total salmoniid biomass. And what we saw is that around 2009, we hit a peak. And if it's interesting because, again, we're still going to continue the study, so things may change. But if we quit the study, say after 2010, it wouldn't look quite as rosy in terms of the sympatry not affecting the brook trout as it does at this time. Okay, next slide, please. Okay, switching from biomass still in the Conway River to look at abundance. So this is a non-significant, slightly declining. I always get these arguments. So if it's non-significant, is it really declining or is it flat? Dan could probably provide some insight later. Anyway, this is numbers per hectare, and this, a lot of this is being driven by year class strength and variable recruitment, of course, with the younger years. This abundance chart does include younger year, but is again not significant trend. Okay, next slide, please. So if we go next door to the Rapidan River, we don't have brown trout, and look at back to kilograms per hectare. Instead of topping out at 60, now we're topping out at 200. Again, a bigger, more productive system. But we have no trend. We have no increase in brook trout biomass in the stream without brown trout. In fact, it's a slightly declining trend rather than a slightly inclining trend. Okay, next one. And we go to abundance, and it's the same thing. It's a wash. But it doesn't appear that either stream is functioning too independently of one another. But what we also did was we wanted to see if over time there was any change, and these are just adults. So this is average size of brook trout in the Conway River. And again, it's one of those things. If we didn't have these last couple of years of data, it would look like maybe average size is increasing. But I think what we're seeing here are some density dependent factors. Because if you look back around 2009 to 2011, we had some of our lowest densities, and we have some of our highest average sizes, probably related to that density factor and the foraging ability of this fish or the system. And the trend line on that was flat, was not significant. It was slightly inclining, but it was not significant. We looked at relative weights, sort of intuitively thinking, okay, if we're adding all this additional amount of biomass, there's probably going to be a decline in brook trout relative weights over time. The red line is the brown trout relative weight line. That is higher in almost all cases in brook trout, which seems to be a normal thing. I guess it's like a European thing, perhaps. I don't know. They're just fatter, nicer, plumper fish. But what we saw with relative weights of the brook trout was that there was no consistent trend. Some years, brook trout in the Conway had better relative weights. Some years they had better relative weights in the Rapidan, and they fluctuated back and forth. So again, there was no trend over time. And again, you see the higher density years with the lower relative weight years suggestive of density factor affecting the health of the fish. Okay. So we weren't expecting this. This is sort of like, wow, this is sort of Buck's conventional wisdom, at least in some cases. Although Jenkins and Burkhead had a citation in Fishes of Virginia saying, and not only what they based it on, but for some reason, brook trout in Virginia appeared to be resilient or resilient against brown trout influence or something along those lines, which I thought was kind of interesting. But again, it was unanticipated. We thought given that amount of additive salmonic biomass, you would see some sort of response. We also looked at non-game stuff, which didn't make it into the paper. Oh, sorry, I got ahead of myself. But to come back to the point I made earlier about the parasites and Mitra's paper. So if you look at some of the more recent literature, Davis and Wagner talked about habitat partitioning and spatial scales. And then this big study in Pennsylvania, Kirk et al., they were looking at primarily at lower elevations. So, I'm thinking what we started to think about was that maybe at these higher elevations in these very pristine systems where there's no environmental perturbations and there's habitat partitioning or there's enough goods for everybody to go around. We also looked at the non-game populations. Both streams had the same populations except for the Rapidan did not have torrent suckers. And we looked at no significant difference over time, although the torrent suckers along those days had some funny stuff going on. And if we quit the study at this point rather than this point, you might have been able to draw some inferences there. But again, I think that's the utility of a long-term data set is we're going to continue to build on this. And we'll get more information. But here's another mouth to feed. Embry Dam came down to Fredericksburg, and now not only does the Conway River have this additive salmonic biomass, but now we've got a lot of American eels up there eating something, right? So you would think at some point something's got to give. So anyway, so at this point, our message from Virginia is hopefully, maybe cautiously, these two may be able to survive in sympatric conditions without an overt negative impact to the brook trout. So we shall know more in another 8 to 10 years, perhaps.
Great, thank you. Any questions?
Dan?
Do you have any temperature data by chance for either system?
We don't. I don't think we have any. Oh, you know anything about temperature data up there, do you?
I think we are collecting some, but
Further down, maybe on the rapid end. Both of these these sample sites are we call the upper sites. So they're higher in elevation. We may have some hobos further down the rapid end, not that I'm aware of. So no, the answer your question, no, we don't have temperature data, but there's no reason to think that the temperature would be limiting at this point.
We're kind of seeing, some of our systems that are pristine and cold that do have brown trout, they have to take an off the brook trout from fine, but other systems that are pristine and warming, the brook trout numbers would tank, the brown trout would come up. So I think it's due to the perturbation, whatever it's called, the temperature start.
And that fits with the Pennsylvania study, the lower elevation thing.
Yeah, we see the same thing, John, in the upper Potomac. We have such a really aggressive gradient. So we go from temperatures that are perfectly suitable for brook trout, no real significant impacts like on our national forest areas, directly into a rock bass, smallmouth bass fall fish stream. And then so when we get all of our anthropogenic impacts, you have a rapid increase in temperatures, which probably blows past that sweet spot for brown trout. So there's no real super good foothold between being able to get up and reproduce super effectively with relatively small numbers and reduce size structures of brown trout.
Right. Interesting.
So,
yeah. That's cool. Quite the transit. Go from one to the other.
Yeah.
No
From
gradient.
national forest circle.
What kind of hydrology do you have of those systems, John? Are they spring fed, not fluctuating, or?
They're headwater streams coming right out the park. I mean, so they're spring fed in freestone, just your typical Virginia Shenandoah non-productive.
do you collect habitat characteristics for those beaches? Those watersheds?
We don't have habitat characteristics for those, no. We do for some. We have some of the BVET stuff for some of them, but not for those.
Matt, we have a question. Looks like a couple questions from Ben. Ben, you want to ask your question?
Yeah, it wasn't really a question so much. It was just we saw similar things in a small stream in Massachusetts.
Great. Thanks for sharing.
Thank you.
Matt Culp, you have a question?
Yeah, hey John. I'm just curious over there, all that length of time you worked that piece of water, how many wild tiger trout did you guys see?
We've never seen any. Now maybe we're not looking for the right things, but to my knowledge, I don't think we've ever seen one in that stream.
I think there's a record of the Park Service maybe picking one or two up in some of their sampling over the years, but I'm not sure if it was in the Conway per se. Because there are other streams in the park that have sympatric brown and rainbow.
Yeah, we've never seen any that look fishy.
The
Thanks.
only places I've seen them in West Virginia is where we have very limited spawning habitat, where they have to use the same habitat to spawn, and their spawning seasons may overlap.
And there's high densities of both.
And there's high densities of both. If you're not spawning habitat limited, or if they partition themselves seasonally, to offset spawning, you may not see pattern trout.
Thanks. We're gonna move on. John, thank you very much. Maybe we can answer those questions in chat or break. So Dan Hitt's joining us. He's next. He's a fish biologist at the USGS, Eastern Ecological Science Center. Dan, come on up. He's working on various aspects of brook trout population dynamics and habitat assessment.
Right.
So good to be with y'all today. Sorry I'm a day late. It's better than my average. Usually I'm 3 days late, so it's cool. So yeah, let's keep this conversation going. What I'm going to talk about today, some experimental work, a little bit of the background literature, and then I'll talk about some new work, some ongoing stuff that's preliminary, but I think casts this question in a different light. So I want your view on this as we go forward. Can I use this clicker here?
It's sketchy.
Sketchy. All right, so here's what we're talking. We all know this conceptually. This interaction between these different elements of physical stressors, biotic stressors, and really the space that we're focusing on today deals with mechanism, competition, predation, temperature, flow, siltation, water quality, this intersection, and particularly with respect to browns. Okay, brown trout, brook trout interactions. That's obviously our focus today. Context. Brown trout have value too. Not news to y'all. So there are these trade-offs. Okay, so let's take a look at the implications for brook trout conservation. We know that we look at the landscape out here. There's a lot of sympatric habitat. Now, at this scale, this is a map you've probably seen before, but at this scale, there's still partitioning going on. We know this, the drill, generally downstream for browns, generally upstream for brook. There also is overlap in many places, like Brandon just mentioned, where we have limited spawning habitat. You have a lot of microhabitat sharing too. That's a real interaction. That's where competition can be relevant. So there's a lot of allopatric habitat, but there's more sympatric habitat out there. The context here is thermal. So the question was raised about the Rapidan and Conway systems on temperature. It's often really hard to disentangle all this stuff because everything tends to happen all at the same time in the same place. So we wanted to use some experimental methods to try to tease out these mechanisms on thermal, and that's where we're going today. There's some good context out there. This URL doesn't exactly roll off tongue, but we did a little literature review, so you guys can check that out, and it'll be in the slides here, just to kind of give an overview of the work that's been done on the topic of brook trout and introduced species, and what are those interactions, what are those environmentally mediated effects of competition, predation, et cetera. A couple key studies, maybe y'all are already familiar with these, but they jump out to me as kind of the lay of the land on this. Kurt Fausch's work in the 80s, an experimental removal of brown trouts. They snorkeled a stream in Michigan. They looked at microhabitat positions. They pulled out the browns. They went back in and looked at shifting microhabitat use. And they found that when they pulled out the browns, brooks shifted their position in the stream towards more velocity resting habitat. So you think about that from an energetics perspective and kind of optimal foraging location position. So that's empirical evidence for competition between these species. And as you know, it's really hard to demonstrate competition, with experimental data. So that's their manipulative approach. That was an important paper in the 80s. Ty Wagner and colleagues, 2013 paper in Transactions, modeling approach from PA. So this is interesting on the elevation question just mentioned, but they used a modeling approach here and looked at species occupancy modeling, and they found that the presence of brown reduced the probability of brook trout presence. So that's an occupancy framework, okay? Implying that there is an exclusion, there's some sort of a competitive exclusion. Now, the effect of temperature in that model wasn't perfectly resolved, okay? But that's the context. Also, Corey's work here, which I think we'll talk about a little later. Dave, maybe you're going to touch on this one. Work from the Shavers Fork and the Cheat River, one of our best connected river networks in West Virginia, cold water river connected system in the downstream areas where you have a lot of effort being placed under, restoring, building pool habitat, building big structural habitat. That's where you see a competition effect with browns. And so again, they did some snorkeling work there in these restored pool habitats. They found that non-native brook trout are displacing non-native browns are displacing brook trout from thermal refugia. So that, there's a thermal effect and resting microhabitats. So there's an energetic effect too. So we're starting to understand something about the mechanisms and also demonstrating experimentally that there is evidence for this competition effect. Question is, well, what's the role of temperature in all of this? And that's hard to untangle from modeling studies. So we wanted to throw some experimental methods at it. So we had a paper a couple years ago in Canadian Journal. We asked a couple of questions. Basically, what's the thermal effect on this competition signal? How do brook trout behave along a temperature gradient in the presence and absence of brown trout? And we used so we're motivated here for this because of some of our work at Shenandoah National Park, where we see a lot of variation within watersheds in terms of this groundwater signal. Okay, the temperature regime, it's not a uniform, it's not a very well behaved signal along the course of these headwater streams. There's a lot of patchiness out there because there's a lot of patchiness in the way the groundwater is intercepting the surface water. And in some cases, like for example, to the southwest here, Payne Run versus to the central part here, Stanton River, these systems are the same size. They get similar amounts of precip, but they have radically different patterns of dewatering. And we're trying to, understand that in some ongoing work, but it has real implications for the spatial distribution of trout in these systems, which basically boils down to the fact that Payne Run on the southwest, dewatering, not every year, but occasionally, you end up seeing much more fragmented populations. So even though the total annual abundance or density of trout can be similar in these places, the spatial distribution of these trout populations can be radically different. So you have all your eggs in one basket, right? Which has implications for extinction or extirpation risk and implications for competition as well. So, this is context, this is our motivation for looking at temperature, because it can be thermally patchy. So we used a experimental stream system that I'm running at the Eastern Ecological Science Center. The system has thermal control. We have some natural substrates in there, recirculating the system. And here's how we set it up. So we experimentally created thermal refugia along a temperature gradient. So we varied ambient temperatures from 14 to 23 C, right at the upper edge, right, pushing the limits of thermal tolerance for brook trout. And then piped in some cold spring water, piped it up through the substrates, the downstream channel. So, you can see, like, basically this would be our headwater channel downstream, then a recirc back at the top. And we wanted to create some variation in the quality of these habitats. So we have foods up here, the red circle, the belt feeder, and the groundwater's down here. So that's a design. And we measured several things using some video sampling. We asked, where are the fish? How spatially even are they? If they're all clustered in one area, how does that change along the temperature gradient? We looked at the occupancy of that groundwater tank, that downstream tank, aggressive interactions, like how much chasing and such, nipping is going on, and then ventilation rate, which is an index of oxygen consumption, just per tone beat rate. And all this was with videos. That is easy to do in a way. So basically, here's a look at the different trials. We've been able to get our target temperatures pretty well, and you can see that on average, we're about 6 to 9 degrees below ambient temperatures during the 23 C, break point. So this really is a thermal refuge, okay? And so we did have patchiness. The quality of these habitats was really different in the stream. So I'm going to show a couple of results plots like this. And you can look at the bar plots on top, but the effect plot on these are standardized coefficients from a mixed model. So basically, this shows us the relative importance of temperature and then brown trout, okay? So what we're looking at here on the top, way to read these, here's each temperature level from 14 to 23 C. And then allopatric brook trout, this is the black bar is basically what brook trout do by themselves. And then S and B refers to sympatric brook trout and brown trout, and then basically the combined, the hatch mark column is basically the combination, the sympatric trials. So the main thing though is to focus on this lower plot where we see, for thermal refugia use, what affects that when we're getting at the bottom, we're looking at brook trout. Okay, what predicts brook trout use of thermal refugia? Well, the effect of brown trout has the strongest impact. In other words, browns in the system, brooks were congregating over that thermal refuge. And the other way to look at this is they were not foraging, okay? The forage was up in the front, the thermal was in the back. And they basically were limited often to that thermal refuge. So they were not foraging in the presence of browns with the same efficiency. On spatial evenness, we see the same signal. It's basically kind of the same story. They're clustered, okay? They're not accessing the forage in the presence of browns, but in the absence of browns, they were. And I'll return to that point here in a minute. On movement, we also saw that there's the presence of browns relative to the temperature gradient. The effect of brown trout was much stronger. Okay, so there was less movement, more clustering, less foraging. And then finally, on aggression, we didn't really see much of an effect there. Aggression was basically the same. And maybe some evidence for increasing rate of nips and strikes and stuff as temperature gets hotter, but no real effect of browns. So brook trout were just as picky on each other as they were on the presence or absence of browns. All right, so last thing, ventilation. And this is a nice clean result. You never see results from a field study like this, but basically, what is it? Brown trout, ventilation rate, no effect. Temperature is the signal. And so this is just keeping up with the treadmill of temperature. Hotter water, more percolate beats, more oxygen consumption. So that it shows us that some of these responses were purely thermal. Others were largely the presence of brown trout. All right, so we can summarize this. For what predicts brook trout responses here on the thermal refugia use, there's an interaction. We see effects of brown trout and temperature, spatial evenness, primarily browns, movement, primarily browns, aggression, no real effect, ventilation, mostly temperature. Okay, so we can kind of unpack some of these mechanisms. Now, what does this mean? It means that there's a thermal effect, right? This is experimental evidence for a thermal threshold by which brook trout use refugia, right? They're not going to be randomly distributed. Even if forage is available, they're going to be going for the thermal refuge. And I think this is the important implication for the competition effect. Brook trout were able to do better in warmer water where browns were absent. And that's the punchline. Because we can't necessarily control temperature. Sometimes we can't, but not often we can't. But we can have more control on stocking programs or on experimental removals of browns or other sort of management activities. And that opens up a couple of options for brook trout conservation that I think are important. So punchline, brown trout, restricted brook trout movement, restricted brook trout foraging. They increased the density of brook trout in these experimental systems. And brook trout did better at warmer temperatures when browns were absent. Okay. Now, this is all fine, and to do it in an experimental stream setting, it's kind of clean, but it's also not very real, right? I mean, we get that trade-offs. So, we can think about how do we move this into a real setting. Now, what are the management implications here? We do see potential benefits for brook trout conservation with the experimental removal of browns, and especially we predict that in the places that are thermally marginal or approaching the thermal limits for brook trout, and this makes sense in a way. These are the downstream sections where you're going to see more sympatry in general. You're going to see higher temperatures in general. It's interesting that the very top of the system, John, you're mentioning, might have been colder. You might see more persistence of both populations in places that are colder. But conversely, you might expect to see the expansion of brook trout. If you could push back browns, they might actually be able to colonize those downstream areas, such as in the Shavers Fork of the Cheek. And that's where you're going to see a lot of growth too. So I think that's pretty interesting from a management perspective. We also, see some confirmatory evidence for this perspective from places like Delaware Gap streams in PA and Maryland. along the Delaware River. Basically, you know, the punchline is that, you know, you can see brook trout in warmer temperatures in the absence of browns. So that's consistent with this idea from a field study. But we wanted to put this to use in the real world. And, you know, this is ongoing work. So this is not a final story, but we see a lot of it taking shape. So I want your feedback on this. But anyway, this is work in conjunction with Maryland DNR up in Big Honey Creek. You know, this is an important fishery in the state of Maryland. John Mulligan and others with Maryland DNR, you guys are managing this fishery for you know, brook trout conservation primarily. So we're talking about basically central Maryland here. It's the extension of the Blue Ridge Mountains up through Maryland. And so there's a waterfall, Cunningham Falls. There's a chunk of the stream here, this Catoctin Mountain Park. And so with Maryland's leadership for the last several years, we've been pulling out brown trout, moving them below Cunningham Falls. So they're still in the fishery, but just moved. And now we're monitoring brook trout, you know, body size, recruitment, changes in abundance. We have some predictions. Do these lab results make sense? Now, I've got to say, the temperature in this system is right at the cusp. This is a place where we would think, you know, this is where we would see an effect if we're going to see such an effect, because ponds that are coming into this site have really roasted the water. I mean, we see, and this is not a Park Service. This is the context of managing Park Service resources or national park resources in a landscape context. If you have water that's on private land coming in ponds, roasted. We also see, for example, a little juvenile bluegill in the streams. It's like, okay, it's a pond, right? But my point is that, you know, this is where temperature is up there. And the question is, can we save this brook trout fishery by pulling out browns and based on our experimental work? Well, all right, so there's a lot of variation. I'm going to show you a couple of plots, time series stuff. That this is Maryland DNR data. Again, this is preliminary stuff. We're not ready to publish this yet, but just for y'all's information. We're going to look at yoy abundances and then you know, adult abundances. And so, you know, there are a couple of different lines here. The allopatric brook trout in orange. This is a controlled stream fishing creek sites in the region, but not with browns. And then the red and blue lines are from Big Honey Creek, and again, above Cunningham Falls. And so, you know, we can see, one thing we can see is kind of some spatial synchrony. You know, if it's a good year for browns, it's a good year for brooks, which is a little precursor, but basically you kind of see that fluctuating pattern. And then basically 2018, we started pulling out browns, moving them downstream. So we would predict that, right? If browns are limiting, then we're going to see browns go down and brooks go up. And what we see here is not that. Now, if you would have asked me a year ago, you know, you cut out a couple of years, I might be like, okay, you know, browns coming down, brooks are going up, but not 2020, not 2021. And I think the punchline here is that, well, I'll get to the punchline, but that's the result for yoys. Let's look for adults age 1+. And look, we've got a disturbing trend line here, okay? And this is one of the motivations for this study. And there's also other background to the stream. I could talk about other fish restoration work that we're going to do there. There are fundamental challenges here, but this is an important, you know, one of the blue ribbon brook trout fisheries in Maryland. And so, you know, we're concerned about this, and Maryland DNR, you know, concerned about this too. So let's pull out browns here. What do we see? Drum roll, please. We see that our reference condition, and there are other streams to look at for reference, of course. Generally speaking, with, you know, bouncing back and forth, but within the sort of historic range. And unfortunately, 2020, 2021. Well, no surprise, the brown numbers are down. We're pulling them out. The surprise is that the brook trout numbers are down too. And so what do I think this means? You know, I think it comes back to temperature. When we look at this site, I mentioned that the temperature is already pretty warm. If you look at the sensitivity of those sites to atmospheric change, this is basically some of our groundwater, surface water modeling. We can build out, you know, forecasts based on temperature and the role of groundwater in creating stability. And the bottom line is that with any increase in air temperature, those sites are very sensitive to air temperature change. So this is our lower stream here in red. Those are all unsuitable sites. That's too hot for brook trout. It's already at the cusp, okay? It's currently suitable. I mean, brook trout can survive there, but it is pushing their physiological limits. And I think what it really means is this is the punchline, is that yes, there is evidence for brook trout and brown trout competition. We see that in experimental studies. We see that in modeling studies. We see that in several ways. But competition doesn't matter when you can't breathe, okay? Because the temperatures here are the primary thing. And we have to think about that from like a physiological perspective. You need to have physiological performance before competition is even relevant. And so, I have to say, I still think that the experimental work we've done here is meaningful. I still think that brown trout management is still a meaningful option on the table, but I also think we have to be clear-eyed about places that may be too hot to pull back in this way. Now, this is an N of 1 study, and we haven't published this work yet, so we're going to be, there's still, this summer is going to be our final year of brown trout removal. But based on the numbers we saw before, I don't think there's any way we're going to see. The only thing we could see this year is a flush of yoys in the system, and that's possible. So we're open-minded to that. We'll see what the data show this summer, but this is work we're going to be publishing next year, and so we'll be following up on this. I think it's important. I mean, it's an N of 1, but it's an experimental manipulation in a place that matters, and I think that the story will be, and I could be wrong here, but I think the story will be that competition doesn't matter when you can't breathe. Some places the temperature is dominant, and that will be a limiting factor. There are other places that are probably warm, but not at that threshold. That would be better opportunities for investment for brown trout and management there. All right, that's what I got for you. Do we have time for any
Thanks,
questions?
man. Yeah, we have time for one question.
Okay. What's cooking?
Hi, Thad. This is Ross Shramko with New Jersey. Very, very interesting study there and great presentation. I'd like to reach out to you, one-to-one afterwards and talk a little bit about a similar situation that we've kind of run into with a brown trout removal and our brook trout response. I think we have a lot of similarities. We certainly saw that as soon as we removed the brown trout, that once those niches were opened and that competition was removed, our brook trout response was tenfold. In a location where we actually thought the brook trout were actually removed from the stream entirely until we did some further sampling and found just a few. So, I mean, I don't have the numbers in front of me, but it was 99% brown trout, 1% brook trout. And as soon as we removed, sorry, there's a lot of background noise here. But as soon as we removed the brown trout, that population has completely flipped. Now we have 100% or 99% brook trout, biomass increases and stuff as well. We're not, I think Sean Krause may have presented this to some folks. It's our Reinhardt stream study. We don't have it published yet. We're not quite there. Small state, small staff. But I'd like to contact you, just get some, just pick your brain a little bit on what we're seeing and how that corresponds to what you saw.
Definitely, Ross, please do Hitt@usgs.gov. And just real quick, I mean, I think we all know the density dependent competition is like super important no matter where you are. The interesting thing is that it's not just the number of fish, but it is the species of fish. So it's greater than just the density dependent. And you expect, hey, where's your biggest competitor? It's going to be an individual that's most like you. I mean, theoretically, this goes way back. It's like you would expect the strongest competition among the most similar individuals. And that's the conspecifics. That's the other brook trout in the system. But yet that's not, it's interesting that you're saying that because that's not what we see, in our experimental lab work. Certainly we saw evidence for competition among brook trout as you'd expect. And, in wild systems, we see that all the time. But the presence of browns had an above and beyond effect that goes beyond just density, which I think that was noteworthy. So yeah, Ross, thanks, and I'll reach out to you as well.
Yeah, Dan, thank you. Let's continue the discussion over lunch and afterwards. There's a lot here to unpack. It sounds like might be some climate change happening right now too with the temperature exceedance. So we're gonna hand it over now to Brock Huntsman. He's a fish biologist with the USGS from the California Water Science Center in Sacramento. Brock, thanks for joining the meeting today, and we'll let you share your screen. He's gonna talk about trout competition in restored habitat.
Yeah, so I'm still disabled for my screen sharing.
You shared Ben's, Gail, you made Ben co-host, you need to make Brock.
Come on.
Okay, there we go. I assume everyone can see
Yes.
my slides. Perfect. All right, well, thanks for having me. So I am a fish biologist with the USGS at the California Water Science Center. And so I'm actually a few years removed from a lot of my brook trout work. However, we did recently publish a paper on the work I'm going to present today. I'm actually glad Dan brought up some of Corey Trego's work. I was originally going to present some of that, but I didn't really have the time to do that. So, but it is really fascinating work, specifically in the Shavers Fork, which is where a lot of this research had occurred. So I was really fortunate to work in Dr. Todd Petty's lab. So I want to acknowledge him. He has since moved on from West Virginia University to Clemson, but during his time at WVU, he established this research program specifically in the Shavers Fork watershed in West Virginia. And so it created this long-term data set that was really valuable for what I'm going to present today. And then Dr. Rhoda ultimately helped me with some of the modeling, so I want to acknowledge him. And then there's quite a few graduate students that went through the program. And so I point out two specifically associated with this work, Eric Merriam and Corey Trego. And then lastly, there's a slew of undergraduate and graduate researchers that help with the long-term sampling and the Shavers Fork work that we've done. So without them, this wouldn't be possible because it is a pretty significant effort to sample a large stream like the main stem of Shavers Fork every year for almost 20 years. And then lastly, a bunch of funding. Ultimately, can't do this work without funding. So Shavers Fork. So Shavers Fork, located in central West Virginia. It's a relatively large watershed, drainage area, the upper Shavers Fork drainage area of approximately 150 square kilometers. And it sits at a pretty high elevation, which makes it pretty unique for the size of the stream at around 1,500 meters. And it's the main stem that's really a pretty fascinating aspect of the Shavers Fork watershed, like Dan was talking about. We hypothesize that the main stem is really important for not only the dispersal of fish among different tributaries in the watershed, but ultimately it results in the brook trout population being pretty well mixed. And so kind of one large fluvial system, which is fairly uncommon for eastern brook trout populations. However, even though it sounds like a fantastic system, and it is, there are still the general threats that the brook trout in Shavers Fork are susceptible to. Acidity is a big issue. The geology of the system has pretty poor buffering capacity, especially in a lot of our tributaries. One tributary in particular where we have long-term data, we've seen historically prior to limestone treatment, that the brook trout population sizes were approximately 1 or 2 individuals, large individuals moving in and pretty much failing to reproduce. And if you compare that to a kind of control site where we didn't have that acidity issue, we typically have a very abundant, kind of healthy population. However, after the restoration and the limestone addition, essentially the treatment site becomes pretty synchronized with the population dynamics of the control site, and get pretty comparable estimates of abundance for that site. So ultimately, one way of treating one of these risks to brook trout in the Shavers Fork watershed through limestone treatment seems to be pretty effective, at least in our headwater streams. Additionally, connectivity is a pretty big issue, even though Shavers is considered pretty well mixed and pretty connected with a lot of the tributaries. Based on some genetic studies, there is still evidence of isolated tributaries in the watershed. And so in 2012, quite a few of these perched culverts that are essentially perched due to a railroad that runs parallel to a lot of the main stem, they were replaced. And ultimately what we see is evidence of brook trout moving into these once isolated tributaries. So again, evidence of some restoration activity in the Shavers Fork, correcting one of these issues. And so lastly, one of the issues that that threatens Shavers Fork specifically for the brook trout population is this issue of warming. And so the downstream reach specifically, as I mentioned, is really important for Shavers Fork. It's not just the corridor, and I'll mention why here after this slide. But in 2012, there was also an effort in the Shavers Fork main stem to do some in-stream restoration in an attempt to not only create more habitat for the main stem, brook trout using the main stem, but also to reduce the temperatures in the main stem. And so there was efforts to create these little cross vein J hooks, as well as these kind of wing deflectors in the river, specifically in the middle reaches of the river where it's pretty wide. It was historically wide, narrow, little, a lot of essentially warming due to lack of canopy and shallow water. So there's a large threat of warming, and we've seen through historic temperature records in these areas that temperatures do reach those thermal limits, brook trout thermal limits, excess of 21 degrees Celsius. So after this restoration was put in place, the reasons for the restoration was to essentially narrow the stream channel, increase the velocity of the stream, create scouring events, and ultimately intercept the groundwater table. The idea was hopefully this would reduce the stream temperatures in these restored areas. And ultimately, that's what we saw. And so this is a paper that Eric Merriam published in 2019, I believe, but ultimately showing that water temperatures in this, specifically in the restored area, we do see this reduction in stream temperatures specifically during those warm summer months. So some more evidence of some restoration in Shavers Fork trying to address some of these threats to the brook trout population. And the reason this is really important, specifically access to that main stem, is that it isn't just a corridor. It isn't just a corridor linking different source populations and allowing fish movement, but it also, we found evidence of brook trout that use this main stem corridor also benefit from productivity. So we, through marker capture studies, as well as stable isotope analyses, we've shown that the fish that use the main stem typically see benefits in growth. And we can confirm that with our stable isotope analyses, showing that typically our largest fish in the watershed tend to have some signature of main stem foraging. So a lot of those threats to brook trout conservation, specifically in Shavers Fork, have been addressed in recent years. But there still lingers this one issue of invasive species, like the previous talks have mentioned, and they can be pretty deleterious to this brook trout using these lower main stem habitats where those temperatures are maybe more suitable for, say, the brook trout or brown trout or rainbow trout than say brook trout. And so these are just some pictures of the brown trout we've captured over the years in different pools within the Shavers Fork main stem. I don't have any good pictures of rainbow trout. I don't know, maybe we just didn't think they were as fascinating as these larger brown trout. But as you can see, pretty large brown trout we capture in the Shavers Fork main stem. And I'll also point out in this picture, this sampling occurred just after the restoration went in. So you can see the boulders from the cross vein that was put in place here. And ultimately, you see how pretty nice brown trout captured from this pool. And so This happened literally a year after the restoration occurred in 2013. So there is this risk of brown trout in these restored habitats, and really the restoration was in place to improve habitat for brook trout, not necessarily brown trout. And so the goal of this talk was ultimately to determine, how much do non-native trout really affect the brook trout's ability to use this restored habitat. And so I'm only going to talk about one of these objectives just for the sake of time today. And so what specifically I want to talk about is a recent publication that we had. It came out either last month or earlier this month, I can't remember. But we published a paper in Restoration Ecology, and we benefited from a long-term data set that we've collected since really the late 1990s in Shavers Fork in the Petty lab. And specifically, we sampled 4 locations, 4 reaches within the Shavers Fork main stem since 2001. 2 of those, site 2 and 3, were in our reference conditions, so upstream of where the restoration started. And 2 of the sites, site 5 and 7, were within the restoration kind of footprint. In 2011, we added 4 additional sites, 1 and 8 for the reference, and 4 and 6 in the restored sampling reaches, because we found out that restoration was going to be occurring in 2012. So ultimately, this set up a really nice Baki type sampling design with before after control impact kind of sampling conditions that we can compare the benefits that this restoration might have had towards the brook trout population. So for our sampling, as I mentioned earlier, it really does take a pretty significant effort to get this type of sampling, especially in a large water body like the main stem of Shavers Fork. Typically, we ran 2 to 3 electrofishing units at a time. Each electrofishing unit, we had a dip netter behind them. We had 2 people processing fish, an individual running fish, 2 processors. But also what we did when we were electrofishing, and also I need to point out that each one of these sampling reaches were typically 800 to almost 1,200 meters in length. So pretty long sampling reaches as well. And in addition to that entire length of stream that we electrofish, we also kept track of what fish were captured from what channel unit. So we also ran a tape and delineated each channel unit as a pool, riffle, or glide, generally speaking. And each fish captured from each of those channel units were then processed at the channel unit scale. So our sampling unit in this case was the channel unit. And so the analysis that we used was a joint species occupancy model, very similar to the Wagner paper that Dan had mentioned, except for we didn't use a direction. We used more of a sympatric model that didn't assume directionality in terms of the competition outcomes. So this is specifically a model that Chris Rhoda developed. He used for some wolves and coyotes, actually. But equally applicable here. And the idea was, can we not only use this occupancy model to investigate how brook trout use this restored habitat and reference habitat in this Baki type sampling design, but also look at these kind of conditional occupancy probabilities to look at whether or not the presence of a non-native trout influenced whether or not a brook trout was present in that channel unit. So here's some of the results. This is marginal occupancy probability. On the left side, we have pre-restoration conditions. The right panel figures are post-restoration. In the top panel, we have our reference sites, and in the bottom, we have our restored sites. The color of the box plots represent different species, and this was a Bayesian analysis, so these are posterior samples. Now these box plots represent the posterior distribution of our marginal occupancy probabilities. On the x-axis, we also have each channel unit that was delineated, so our sampling unit. And so there are 2 general patterns I really want to point out about these results. One, at the channel unit scale, we saw that brook trout, regardless of the before-after control impact kind of or Baki sampling design, the treatment type, at the channel unit scale, we typically saw higher occupancy probability by the brook trouts in riffle habitat compared to pool or glide habitat. And this was consistent whether inside or outside of restoration, before or after restoration occurred. Alternatively, the non-native fish, both non-native fish, brown trout and rainbow trout, typically occupied those deeper pool habitats in higher proportion than they did in the other two channel unit types. And again, this was consistent across all the treatments. And then the second pattern I really want to point out is that there's really seemed to be more of a temporal pattern in the effects to occupancy probability for all three species as opposed to a spatial effect. And so we found that pre-restoration, we typically found a lower occupancy probability of our brook trout in all the channel units types compared to post-restoration. Occupancy tended to increase post-restoration. Now this occurred in the reference and the restored sites. Alternatively, the non-native fish showed the exact opposite. So the non-native fish occupancy probability in all channel units actually decreased from pre-restoration to post-restoration. So pre-restoration occupancy probability was much higher than it was post-restoration for the non-native fish. And so this suggests that the restoration may not have had as strong of an effect on the occupancy or the use of main stem habitat by brook trout as the decline in the non-native fish in the main stem. And this is mostly due to the lack of an interaction between the spatial and temporal component of this Bocce design. Additionally, to really further investigate that kind of potential competitive interaction and displacement by non-native fish on the native brook trout, we looked at our conditional occupancy probability values. So on the x-axis in this plot, you see the probability of brook trout occupying a channel unit conditional on brown trout also occupying that site or that channel unit. On the y-axis, we have brook trout occupancy conditional on brown trout being absent from that channel unit. And this one-to-one line ultimately reflects that points above the one-to-one line are where occupancy probability was higher in habitats when brown trout were absent versus when they were present, which would be below the line. And so to no surprise, what we found was in all three channel unit types, brook trout were more likely to occupy habitat when brown trout weren't present. So obviously, no surprise, I would assume to anybody. And this was specifically, or most obvious really in the pool and riffle habitat. The glide habitat was really just poor habitat all around. So typically we didn't see many trout in glide habitat regardless of the species. So if we do the same thing with brown trout and see what the conditional occupancy probability looks like. So on the x-axis, we looked at the brown trout occupancy conditional on brook trout not occupying that same channel unit. And then on the y-axis, brown trout occupying a channel unit that was conditional on brook trout also occupying that channel unit. And again, the same interpretation of the one-to-one line. And so I think this plot is actually a little more interesting and revealing about what really is probably going on with the brook trout occupancy and habitat use in the main stem. What you see specifically in the habitats occupied by both brown trout and brook trout, that there really doesn't seem to be any use of any channel units by brown trout when brook trout are occupying that habitat. You can see this; these are essentially just representing the distribution of that occupancy probability, conditional occupancy probability for each of the three channel units. And so there's a lot of overlap there. You see ultimately low occupancy probability when brook trout are present. But when you look at when brook trout aren't in that same channel unit, you do see a strong separation in what habitat the brown trout really are selecting for. Specifically, they really like those pool habitat. And so what if you look at face value, this would suggest that the brook trout affect whether or not brown trout are actually occupying a channel unit. Obviously, I don't believe this is actually the case. I think it more likely reflects more that the brown trout really prevent, at least in Shaver's Ford, prevent those brook trout from really using the channel units that they prefer to occupy. And some of the snorkeling surveys that Corey Trago had done in his 2019 paper, also published in Restoration Ecology, really shows that those brown trout really prevent, especially smaller brook trout, from using the same channel units, or especially those pool habitat in the main stem. So, but however, the larger brook trout tend to also occupy similar habitat as the brown trout and rainbow trout. So kind of looking at this idea that those larger fish, larger brook trout may be competitive enough to occupy similar habitat where the inferior, smaller fish are forced into marginal habitat. So summary, ultimately, I think it's pretty obvious that the presence of the brown trout in the main stem does have an effect on whether or not the brook trout are using that main stem. And again, that main stem is really important, not only as a corridor linking these different source tributaries, but also as a foraging habitat that can result in larger, more productive brook trout. I think we really benefited from this essentially Baki design that we had, which was really only possible because we have this long-term data set that was developed from a slew of different graduate students that worked in the lab for almost 20 years. I think it was really interesting that we just saw a temporal pattern affecting the occupancy probability of the different trout species. And I think this temporal pattern being that the higher occupancy probability by brook trout post-restoration, but lower occupancy by non-native trouts post-restoration actually reflects more that the brown trout and rainbow trout really, we don't see much evidence of reproduction or recruitment in Shavers Fork. There is a little evidence in some of the larger tributaries, but not a significant amount. And these declining numbers of non-native fish on top of the lack on top of stocking no longer occurring for the brown trout in these upper regions of the upper Shavers Fork likely is responsible for this kind of recovery of the brook trout in the main stem. And so maybe following Dan's talk where he mentioned that, sometimes there are these other factors that maybe mask the benefits that could be experienced by removing the brown trout. We actually do see that as those non-native trout, and specifically brown trout, densities and abundances decrease in that main stem, we have seen evidence that the brook trout are tending to have a higher use of those main stem restored habitats. So again, we can't just look at one factor, what's setting these carrying capacities and are we at stressful limits, but also that those brown trout are important for affecting whether or not those brook trout are using those habitats when we're within those suitable conditions. And so that's everything I have.
Great, thank you, Brock. Questions? We have time for 2 questions.
Kind of a general question, and it might be more suited for a geneticist if they're on the call, but thinking about the Shavers Fork and where it's located geographically, being high elevation, and you move that north, it's almost, correct me if I'm wrong, if it's kind of a Canadian stream where we can grow big fish. Have we lost, the genetic component in the eastern part of the U.S. for brook trout to grow to bigger sizes that we see up north? Is that why we're not seeing these restoration benefits for brook trout? Because they're just at a size disadvantage in general?
Yeah, I can't really speak towards any of the genetics component, definitely outside of my wheelhouse. I know, looking at just kind of some of our general genetic studies that have come out of here, there was a paper by Noonans et al. 2015 that looked at the genetics of Shavers Fork and a few neighboring watersheds. It definitely showed that the brook trout and Shavers are more well mixed. There's not these really distinct kind of genetic signatures in a lot of the tributaries, suggesting there's a lot of movement. So it does seem to be, at least Shavers Fork, kind of an anomaly in that that main stem, and the hypothesis really is the reason for that well mixed, that mixing is that main stem, there's reasons for those fish using that main stem to experience benefits outside of just movement, but also somatic growth is one of the hypotheses for why we see that. But as for the rest of your question about the genetics and the loss of this kind of genetic signature and this kind of fluvial life history state, if you want to call it that, I can't really speak towards that.
Okay. Well, thank you. Thanks, Brock. Please follow up with Brock with any follow-up questions. All right, we've got Matt Culp. Many of you know him. He's been a longtime member of the steering committee out of the Great Smoky Mountains National Park. Fisheries biologist there, and Matt is going to share with us a presentation on non-native removal and brook trout reintroductions, review of past projects and examples. So we're making you a co-host, Matt. You should be able to share your screen. Thanks.
All right.
All right. Great.
You guys got that?
Looking good.
All right. Well, thank you all very much. I wish I was there with you guys and gals. I miss seeing you all, but appreciate the opportunity to speak today. I want to thank Lori and Nat for the opportunity to speak today on this. And also, more importantly, and equally important, I guess, is just to thank all the co-authors. This was truly a team effort, and it was basically a summary of all the work that people have done in all these states and federal agencies. And so I want to give kudos to all these folks. I also want to say thanks in the sense that all of them got their information to me before the deadline we put on so we could present this today. So real professional group. If your name is on there, I really appreciate all your help, and you're an equal part of this effort. So really the purpose of this today was to just summarize the history of brook trout restoration with regards to the use of fish toxicants, the different electrofishing techniques, and translocation. And when I speak to translocation, that's not just moving fish into fishless places, but also translocation after either liming or maybe even a culvert removal type of project. And the temporal context is we're going to focus on work from the 1990s to present, and talk about those projects in the more recent times. As far as the spatial scale, we have projects represented in this from Georgia to Maine in the typical eastern portion of the range, as well as projects in Ohio and Wisconsin. The other thing I want to note is that it also includes, I think, all the life history forms. We definitely have lakes and ponds and streams, and I believe we might even have a salter population or two represented in this data. So again, thanks to everyone for their contributions. We saw this earlier, and I wanted to note that a couple things that we've heard the last couple of days. We all know about these major threats that were listed here in the 2006 status and trends, and typical ones that we see. But you know, those non-native species are a big part of this. Odenkirk pointed out not just non-native species, brown trout have their own listing here. But I would go a step further and say that, you know, even in the more recent regional threat assessment that was done by the joint venture, the one that sticks out and this is listed for each of the three regions were non-native species. And so I think, you know, anything we can do to restore populations and remove non-native species and put brook trout back is a big plus, and you know, certainly something that is a priority for each and every one of us. The other thing I want to mention is that, you know, this was mentioned as well in these conservation actions. And when you talk about restoring brook trout back to their former habitat, you're really hammering away at all 6 of those, in each in their own way. And so again, this is, I think, one of the most important things we can do as managers of these populations is to look for opportunities to restore these things where we can in these different variety of ways. And so before we jump into the contemporary work, I thought it was worth noting some of the historical work. There's not a lot out there, you know, on the landscape prior to 1990s. However, there's a couple states that were doing, one in particular here in New York, that definitely deserves kudos. I think New York was definitely ahead of their time in the 1950s all the way up through the 1970s. And the fact that they restored, you know, upwards of 125 lakes and ponds by 1975, and even constructed some barrier dams. And, you know, if you think about the histories of fisheries management, I think that was really progressive for that era of time to be thinking about native species restoration. There's definitely some other states and some national parks and federal lands like national forests where they did some other work. Some of it not so good, you know, using rotenone to remove what they call trash fish. and even some other techniques were used to restore trout, but they were met with mixed results just because of the inefficiencies, capture inefficiencies back in those days. So let's talk about the data from this study, and got it summarized here in a table. And basically for each state, agency, or national park, it has the number of projects on this table, and right below that, it has the percent of those projects that were successful. And so a couple takeaways from this. First of all, 15 of the 17 states in both the national parks have conducted 191 projects over the last, say, 30 years. And what's cool about that is that's increased 36% just from 2018. And so there's been a number of new projects that have come on board just in the last 4 to 5 years. Rotenone was the most highly used technique. And most of those projects were in New York and Maine, and on ponds and some lakes as well. The next most used technique was translocation, and was closely behind the use of rotenone. And again, those included not just translocating fish into fishless areas, but also there's been a lot of recent work, especially in West Virginia, with locating fish into streams that have been limed to restore that habitat. We'll talk about those more in a minute. Then the next two had to do with electrofishing, multiple electrofishing removals, and then annual electrofishing removals. They were the next two most prevalent techniques. And generally, the multiple electrofishing removals had higher success than the annual removal projects. So I want to give a sense of those projects across the spectrum of the states and national parks as well. And we'll look at these from the 2018 data to the 2022 data, and they're fairly similar, but I wanted to point out some differences from 18 to 22. So here's our 18 data, and here's our 22 data. And similar patterns, but a couple of things worth noting. First of all, again, most of the projects were in New York. A lot of those, again, were those lake and pond projects. But I think it's worth noting those with the stars. Those are 3 states that added projects just in the last 4 years. They're in Pennsylvania, Maryland, and West Virginia. So kudos to those folks for taking on some work and getting that stuff done. Those percentages you see are percent increases since 2018 in terms of the number of projects. And so I think the other thing to point out is there was a lot of activity, especially in those southeastern states. And so kudos to those folks as well for taking on those projects. And the other thing I think you'll note in those southeastern states, there's a lot of diversity in the types of techniques that they're using as well. The next thing I wanted to point out was the stream kilometers restored, or the hectares of ponds or lakes restored. And again, a lot of hectares in terms of ponds and lakes restored in the northeastern states. And the transition into 2022, it pretty well holds the same. New York still has a lot of ponds and lakes that they've restored, and they increased that by about 3% over the last 4 years with the lake addition that they had. Then you've got all these southern states that are noted here that have really done a lot of work and increased their stream kilometers quite a bit. I think notably, that West Virginia, they put a lot of stuff on the landscape that they had done, as well as a couple of those other states that you see there. Generally, there's fewer projects that have been done across the Northeast, but a lot of those, again, are in those ponds and lakes that were done historically. You would also note here that 2 of the states, Ohio and Georgia, actually lost some kilometers, and that was done because of reassessments of those projects, and they found that they just weren't successful. And so in some cases, there was a little bit of range loss as well, since the last time these states were looked at. So of these projects that failed, why did they fail? And it really didn't change a whole lot from 18 to 22. What we saw was these chemical treatments or toxicant treatments, all of them failed, the ones that failed due to insufficient treatment. And so whether that was, either wasn't applied thoroughly enough, or there were some areas missed, or some cases even a bad product, that was the leading cause there. In terms of translocation, the number one reason was unknown, but my guess would be if they truly knew what the issue was, it probably would be related to the other 2 listed there. Poor fry survival and poor habitat would likely be the case. And then for the electrofishing projects, really the 2 main were ineffective barriers and failure to remove the non-natives. And I would just note for those folks thinking about doing this work, there are ways to evaluate those barriers using what we call a mark and move survey. And if y'all like to hear more about that, myself or others can talk to you about that as a pretty good means to test those barriers prior to thinking about treating something. We're able to also look at cost, and we tried to do this. It's kind of difficult, and just take this with a grain of salt, and that this data is kind of apples and oranges, and that some states and federal lands projected cost per kilometer based upon just the treatment itself. Some included pre-treatment versus post-treatment, but I think we're able to get a little more clarity with the 2022 survey because we got a little bit tighter ranges. These means basically show that the most expensive per kilometer was the multiple electrofission removals, which you might expect to be the case given the amount of effort you've got to put in putting numerous removals in over the course of 1 to 2 to 3 years. Second most expensive per kilometer was the chemical treatments. But again, the thing to note with those, many of those also included the pre and post monitoring as well that was factored into there, and the staffing for that. The cheapest, as you might expect, per kilometer was the translocation. If you've got a piece of water that you can just move fish into, it makes sense that it doesn't cost as much just to move fish into those areas. And so that was the cheapest that we saw. How about the reasons for failure? We move on to the public perception. So what did the public think about these things as well? And these didn't really change from 18 to 22. In most cases, the public thought they were favorable to highly favorable for all these different projects. And in many cases, it was hard to assess public perception, as some agencies had little to no reporting of the public perception on their projects. So it was relatively unknown. But my guess would be, had they known about it, it would have been looked upon favorably. And just to dig into that a little bit more, the public perception of these, there's some examples I think we can throw out from some state and federal agencies here. A couple chemical projects I found interesting years ago. One of the chemical projects in the Smokies, that people weren't really opposed to the chemical treatment itself. They were opposed to replacing a perfectly fine rainbow trout population with a brook trout population. And we've heard that from some of the state partners as well. And then there was a second project where we had a unique situation. Despite the favorable nature of the project, there were some people that just wanted to stick it to the government, so to speak, and they sabotaged the project. And we're able to go back and salvage that project with retreating the lower end of it. But the point was, there had to do some work to explain to folks how this is part of their heritage, and these fish are native to this landscape and important. And that was summarized in a recent chapter in a book from Catastrophe Recovery and an AFS book that kind of highlights fish restoration across the country. And so I encourage you to check that out if you're interested. Some other perceptions from the states. In many cases, states had mixed bags. A lot of the positive perception was had to do with eradicating non-native species. Those states like New York that use the toxicants, sometimes people get hung up on words like poisoning or toxicant and get hung up on that. But for the most part, for other states and that we've and federal agencies, most of it is favorable. And some states purposely flew under the radar in not reporting some of their data, which I think is really a mistake. I think when you look at Wisconsin and North Carolina here, what they found is that from the anglers that were aware of these projects, most of them were very satisfied, and over 75% were satisfied here in Wisconsin. And in North Carolina, I think to kind of put an icing on the cake, more recent data from Jay Grash down there, when people were asked about brook trout restoration, it was of high importance to their trout anglers. So again, I think, not to run away from putting this out to the public. I think the public is actually, in most cases, pretty supportive of this type of work. They understand why we're doing it and the benefits that it'll have to them. The last thing I wanted to share was that, I was able to go back and just look at a literature review of journal articles specific to, restoring brook trout populations throughout the range and using these different techniques. And I think there's a few interesting things here to look at. First of all, we can only find about 39 articles that directly related to brook trout restoration over the last 63 years. And I think, first of all, that's an opportunity for all of us to do a better job of publishing some of this type of work. It's not necessarily something that fits in the literature well, but I think there's some places that we can get it out there and help each other out with this type of work. Second thing is most of the articles that are out there have to do with some of the liming work and the restoring populations with liming. And a lot of that peaked in the late 90s, but some of it is still being published today. More recently, you're starting to see more of these articles on genetic rescue and culvert removal, and even things like using hatchery propagation of native fishes, to reintroduce native brook trout back on the landscape. But again, I think a lot of work and some challenges here for all of us to get this stuff out in the published literature. So to summarize, again, people should be proud. There's a lot of the states are working on restoration projects as well as our national forests and national parks. And so really important work. And I think it's great to see that work being done. Again, rotenone was the most highly used, but translocations were a close second and growing, I believe, and something that we're going to continue to see more and more of. And those are followed by the multiple removals and the annual electrofishing removals. The cause of failure, changed for each technique, but for anamycin and rotenone, it was really insufficient treatment. And then for translocation, probably poor fry survival was the #1 reason. And for both electrofishing types, it was really that failure to remove the non-natives and the ineffective barriers. Again, in terms of the public, I think most of these are highly favorable with our public if they're aware of them. And so we shouldn't hide our heads in the sand on these. We should let people know what's going on. And I think we get a lot of support for them. And don't be our own worst enemies on these things. Let folks know and put it out there. A couple last things to mention. I think there's a lot of potential that we've heard about already, especially now that we're getting these catchment mappers updated. There's a lot of potential out there to restore populations, and people should go after opportunities to do that. The joint venture is one great funding source. There's many others out there, and having sat on those scoring panels, every one of these projects that we see come through to restore brook trout, the native habitat typically scores very well and usually gets funded. And so you need the seasonals, you need interns, you need, some assistance, put it in there, and go after these things and get with your partners and get some of these things done. And then finally, again, this type of work helps meet not just the conservation strategies of the joint venture, but fits perfectly in a lot of your all's fish plans that you talked about yesterday. And I think is one of the best ways that we can help restore these species across their range is to get some more work efforts in these restoration techniques. So again, thank you to all the folks that contributed, and really appreciate all your help. Thank you all very much. And I will provide this table and this presentation back out to everyone that participated, and so you have that for reference as well. Thank you all very much.
Thank you, Matt. And thanks again for all the work you put into summarizing and analyzing the data that everyone provided. It's a lot of great information. So questions for Matt? Plenty of time for questions. Steve.
Matt, Steve Reeser. Just as kind of a question to you and maybe a comment to the whole group about your thoughts on maybe the future with introducing YY or tetraploid non-natives to brook trout watersheds as a way of restoring brook trout and removing non-natives.
I think we need to try it. If you talk to Dan Schill and Kevin Meyer, they're going to tell you need to thin out, those non-natives first and then put those YYs in there. But they're looking for places in the East. So if y'all have a likely candidate to try that, I think the work they've done so far would say it's definitely something we should take a look at for certain places. I don't think it's the holy grail for everywhere.
Thanks, Matt. Appreciate it. Jason Coombs. I just had a question on the translocations. Maybe this is more a question for the states in general. Are you guys taking genetic clips of the fish that you're moving, or maybe not running them at this point in time, but having them for reference if you were going to do a genetic monitoring? I don't know who wants to answer that.
I'll just start by saying that for us, we do in some cases, Jason, but not in all. And we didn't ask that question in the survey, so the other states have to jump in.
I mean, you're handling the fish anyway. That it's never a bad idea to have this, even if they don't get run, you just put them in an envelope and leave them on your desk. And then maybe somebody down the road will want, but you'll never get a chance to get that information again at that point in time. I just seen that there is an increase in the number of translocations going on, so it's something to think about when you're doing it. Which
Jake,
is-
for all the brook trout that we've run through Raymond Farms, which is probably approaching over 3,000 now, I have fin clips for it. And all of them have been leveraged into either genetic rescue and more specifically repatriation issues.
Yeah, because we're gonna want to address a lot of this, the outcomes of these translocations. If we can get the genetic component of that to really tease it out, that'll be really important to understanding how this management action affects, our actions.
So
I
we
agree,
have
Jason.
totality of the gene pool that was utilized for that.
I will say, Jason, too, that in most all those streams, the reason we are translocating them, at least in the park, is because we know what the genetics are of those source stocks. And so we're not just moving stuff without that, but we don't always have this genetics of those individuals that we moved for that particular translocation event.
Yeah, this is Ross again from New Jersey. That the genetics or lack thereof in New Jersey, the work that we've done with genetics is extremely little. That's been one of the reasons that we haven't done translocations or any type of obvious genetic rescue or places that we know have been isolated due to dams for over 100 years, because we want to get that genetic component first. I mean, I know sometimes we shouldn't let those things, completely stop us. And like Jason said, we could certainly store the samples, but even, in a state like New Jersey where our source populations are also limited and at risk, we don't want to just move something and not know why or be as efficient as we could be. So certainly I think, the genetics or the know-how is so important, but it's actually slowed us in our brook trout restoration efforts.
Yeah, makes me think of some discussions, Jake, that we've had. I know that was one of your plans for North Carolina. I don't know if you want to weigh in on that topic.
I can, if I can figure out how to work my computer. Yeah, we've been attempting to, clip everything that we get our hands on, just, for the future, to be able to circle back and, kind of like you're saying, Ross, to measure our effectiveness, not just in the short term, but the long term, and glean as much as we can from, sort of these genetic based approaches to designing, implementing, and then ultimately trying to monitor the outcomes of our restorations.
Thanks, Jake. Any more questions for Matt?
Yeah. Jeff Day here in Massachusetts.
Hey, Jeff.
Yeah, thanks for taking my question. So here I'm in northern Mass, but I work primarily in Southeast Mass, Cape Cod with a sea-run brook trout. Does your data, I saw some data from Massachusetts, does your data include 3 rescue situations down on Cape Cod with salters?
I've got 3 populations for Massachusetts: the Childs River, the
Quinnemessett and Sentuit.
And the Sentuit. That's correct.
Excellent. Okay, fabulous. That's all I wanted to know. We are also concerned about heterozygosity, and I need to ask if there's somebody there that I can talk to about heterozygosity offline. Would that be Jake?
I say Shannon White's on here. She'd be excellent.
Shannon, okay. I'll talk to Shannon. Thank you. Excellent.
And speaking of Shannon, that thing you just brought up, Nat, about follow up, Shannon and Dave Kazak up there at the Science Center have put in for a project to do some follow up genetics with, I believe, maybe Mariah Meek, to look at the source stocks that were put in a lot of these populations anywhere from, 2, 3, 4 years ago, all the way back to 25 years ago, and to see what those populations look like now, to look at differential selection, to look at a whole range of things as far as how many initial source stocks were put in from each contributor. And so I think that's a really big piece of work that, Jake's all of us are really interested in to see, to try to learn from our restoration techniques and improve, how many, how often, that kind of thing. So she might could speak to that as well.
Thanks. Shannon, do you want any comments?
Yeah, we've been doing a couple of different proposals looking at both range wide, but I think mostly focused a lot on kind of southeastern populations, especially with Jake and Matt, trying to really get some of these historic data sets to start looking at some of the effects of reintroduction from that genetic perspective. I think we've talked to a lot of you over the last year or so, trying to just really get some momentum to get some samples and to start answering some of these questions. So it'd be a great opportunity for collaboration for sure.
Oh, Jake, do you have another question?
Yeah, and apologies if I whiffed on your question earlier, Nat. But thinking about this stuff, but I appreciate the follow-ups on the phylogenomics work that hopefully we can track down to help us dive deeper. But I also want to revisit Steve Reeser's question about this YY super male deal. I think there's enough interest, at least in our area, to do this, and we would do it if we could grow the fish. And so I don't know, maybe that's something we can all get together offline in this geographic extent to see if we can't maybe identify somewhere to help us produce those fish. Our major facility where we would do that is going to be under renovation, so we're kind of out for the next 5 years. And so I don't know if there's a federal facility that might be ideal as kind of the central hub to help us get those lines going, to let us in the States, start to experiment and try that work here. So just kind of throwing that plug out to have us maybe talk amongst ourselves and see how we can try the, push the pedal on that to get going.
Thanks, Jake. Fred, couple comments in the chat. Do you want to share any of or both of them from New York?
Sure. Just to sort of tangentially related to the translocations is that we've had a number of ponds in the Adirondacks that have been naturally recolonized by brook trout because the habitat is recovered sufficiently from the acidification so the restoration, the mechanism of restoration was really the Clean Air Act. It's not hands-on in the sense of us rote-noning or liming, but nonetheless, I think that is a restoration mechanism that shouldn't get lost. My other comment was just in terms of was to Jason's question in terms of the source populations that we use for our restoration work in Adirondack ponds. We have good genetic background at the population level, but not on the individual fish that we're stocking. And we also maintain disease histories on those populations.
Thanks, Fred.
Yep.
Yeah. So the great topic. I would say if folks have questions for any of the other speakers during this sort of panel, bring them up and we can hopefully get them answered.
Fan, question about your study, your living stream study. I assume that you use pretty similar size distributions of brook and brown trout simultaneously in the facility.
Totally. So the body size was standard, which is, again, artificial but controlled, you know?
Right. Yeah, because that gets a behavior rather than size dominance.
For the Corey's work and the shavers, I mean, you know, it's not a surprise that body size is just obviously blown up for browns, but in our system, to control for that. These are hatchery fish too. So that's another asterisk.
With respect to the influence of temperature on competitive interactions, there was some interesting research published by John Hoxmeyer out in Minnesota, which showed the kind of the unusual situation of a sympatric population where over time brook trout displaced the brown trout that had invaded the system. And I believe he had some data showing the temperature, groundwater influx had increased over time, and he was attributing to that. Are any of the authors who presented today familiar with that research and maybe comment on it in light of their own findings?
Yeah, Fred, real quick. I mean, and maybe John's on the call here. I don't know, but my take from their work, there was a few studies came out of that lab, and one of them showed landscape level benefits, and they saw a natural effect. Then they also, if I'm not mistaken, they had manipulated brown, so they pulled brown numbers out and saw a real increase in brook trout body size and recruitment. So I mean, they saw at the stream scale, you know. So that work pops out to me for the groundwater story, but also, you know, it was a restoration success story. But again, these are sandy systems loaded with groundwater, and that's part of the mix. And they're low gradient systems, but they're really dominated by groundwater. Yeah, the Hoxmeyer lab work is really notable for this.
I wonder too if brown trout aren't able to get a better hold in downstream reaches and develop more fecundity in the form of big females that they use to swim upstream and spawn for recruitment, and systems that have higher species richness when it comes to fish because of that transition from an invertebrate to piscivorous, and that giving them the ability to just carry just gigantic volumes of eggs around and displace brook trout through recruitment.
Well, it's interesting because I think Brock Huntsman's still on the line, and their work in Shaver Sport, and maybe Dave, you've seen this too, but it's brook trout that are moving upstream tenfold over brown trout moving upstream. Absolutely. Okay, so the movement signal is a brook signal. The connectivity at the stream to main stem is brook, not so much brown.
I was under the impression that brown trout also use those smaller streams, that either the mouths of them or even the main stems of those smaller streams to spawn.
Yeah, Brock's on. Any thoughts there?
Yeah, so in Shavers at least, we did have two tributaries where we found evidence of essentially young of the year brown trout. They're pretty large tributaries, but we never really saw a high number of them. And it was actually, I believe, now granted, I only did 3 years of field work while I was there. So I don't remember in our data ever seeing a lot of young of the year brown trout in those tributaries. And yeah, the really that benefit that we often saw for the main stem foraging, at least in Shavers, was, you know, really focused on those brook trout and finding those brook trout in those tributaries with that main stem signature. And a lot of that was based on some of our isotope work. But we never actually used or tested brown trout, but we rarely saw, you know, outside of some of those like kind of near the confluence of some of those larger tributaries, we never really saw brown trout in them.
Does that include young of the year as well, Brock? I know it's a low density brown trout population, so a couple streams worth of productive recruitment was really all it would take for them to, you know, filter back downstream and then occupy the main stem and have virtually 80% survival until they become 24 inches long.
Yeah, well, so in those tributaries, yeah, definitely are well, I shouldn't say definitely, but we've seen a few instances of those young of the year for sure. Now I've rarely seen like a large brown trout in those tributaries, but again, that mostly we find those larger brown trout just in the main stem. Rarely see them in the tribs. But I should also point out that most of our sampling typically was done in June. So we weren't up there when the fish were spawning.
And this would be a situation too where brown trout aren't displacing brook trout from those small tributaries either.
But you gotta tag them to see where they're going, if they're moving, how else.
I would just say that in our long-term study site in Massachusetts in the Westbrook, we hardly ever saw brown trout use tributaries to spawn. It happened a few times, right, in very close proximity to the main stem, but we hardly ever saw them spawn in the tributary.
I agree, and just anecdotally, the only spawning brown trout I ever saw in shavers were in the main stem. There were very few incidences of brown trout at the end of the year in the tributaries. Rainbow a little bit more, but the rainbows really didn't encroach very far upstream. It's generally acidic system, so the rainbows aren't going to encroach into those more acidic tributaries.
Do you want to answer the question for Andy?
Yeah, actually, yeah. Hey, Andy, nice to hear from you. Regarding your question about the Rapidan brook trout after the epic Madison County floods of 1995, that was the lowest biomass ever recorded in the Rapidan River. I think it was 97 or 98, but it rapidly recovered.
John, I really love your presentation. I'm just thinking out loud if there's other St. Patrick sites that you or other colleagues in here are interested in studying to see, are there some characteristics that would lend to where brook trout do hold their own, apparently with brook trout, because we all know of those systems where they've been there a long time, coexisting more or less, we think. And why is that the case?
In a follow-up, somebody asked me before about thermal effects. We do take temperature every time we sample. We just never modeled it because it didn't seem to be an issue. But we can go back. I mean, maybe in the last 20 years it has changed, and maybe it will change in another 20 years, and that may be in effect. So yeah, to revisit that, absolutely.
I got a bit of a weird one. Probably our best wild brook trout fishery in Massachusetts, the bottom release tailwater fishery. Upwards of 3,000 brook trout per kilometer, up to almost 2 feet long. They're big fish in there. It's also the most heavily stocked water body in the state, rainbows and browns, broodstock browns, and it's just blanketed with brook trout. But it's essentially stays the same temperature year round. So
what is the temperature?
It's between like 13 to 14 degrees. And so, I mean, the browns try to reproduce, but I've seen one young of the year brown trout in like 5 years. So, I mean, I don't really know why, but it might be a temperature that the brook trout just hatch out that much earlier, and blanket the habitat and nothing else can get established.
Any other questions? Okay, that was a great morning. We're gonna